Hello, everyone. I hope you all are doing great and welcome to the Science Prism once again. In our previous session, we had talked about genes, we had studied what is genetics, we had talked about Mendel's experiments, and much more. Now, in today's class, it's time to dive deeper into the topic, and I'm sure you are finding this particular unit very, very interesting. And believe me, this unit has a lot of conceptual things, and once you understand, it's very scoring also. So, moving ahead to the video that I want to show you. It's a brief video because in today's class we'll be talking about something interesting in which we'll be talking about genotypic and phenotypic ratios, right? For that, I want you to help you so that you can revise what is genotype and what is phenotype.
Confused between genotype and phenotype? Geno means genes.
So basically, geno refers to the genes. For example, if you are taking TT, capital T, small t, or both small t, which will refer to the dwarf plant, whatever genes you are talking about, they come under genotype.
Like big T, small t. Pheno is the physical appearance, tall or dwarf. Confused between
Okay.
genotype and phenotype?
So the physical appearance that you observe, whether it is the color, whether you're talking about the color, whether you're talking about the shape of the seed, or you're talking about the the tallness, tall or dwarf, that all physical appearance comes under the phenotype. Okay, so today we'll be diving deeper into it because we'll be talking about monohybrid cross. So when we'll be discussing about genotypic and phenotypic ratio, I hope you'll not face problem in that. So now, moving ahead to what is a monohybrid cross. Now, basically, monohybrid cross is the study of inheritance of one pair of contrasting characters. What do I mean by one pair? I think I'll write here. So I'll switch off the video and I'll mention, because after this we'll be drawing the cross also, right? So I'll make the complete space on the screen for you. Okay. I hope it's like completely only the screen.
Confused between.
All right. So when we study, when we talk about only one pair of contrasting characters, now what is the meaning of contrasting? The word contrast refers to the opposite traits. Okay, the opposite traits which are passed from parents to their offsprings, that study is called as basically a monohybrid cross. Now, in monohybrid cross, then we look at one pair of character at a time. For example, just see this. We are talking about tall and dwarf. Now, what is the observe? What are you going to observe? What are the features? One is a tall plant and another one is a dwarf plant. So here you have two opposite, that is two contrasting characters. Okay, one tall, another one as the dwarf. Similarly, let's take the example of curly hair and straight hair, two opposite characters, two opposite traits we are talking about. But there are two terminologies that will help you to to know the chapter more in detail. That is one, dominant. Now, dominant is the one that expresses itself, the character or the physical appearance that you observe. The other one is the recessive, which might be present, but maybe it is hidden. Okay, for example, capital T and small t. In this case, what will be the physical appearance you observe? You'll see the plant will be tall, but at the same time, this gene is also there, which is recessive, and it is recessive, that is why it is hidden and it did not show up. But the plant is tall because of this particular, which is dominant. So basically what happens is, in a monohybrid cross, we talk about the study of inheritance in which one pair of contrasting characters we will check. Okay, so now we will draw a cross so that it will be easier for you. So let's see here. What is happening in this? Mendel basically cross-pollinated homozygous tall plants with homozygous dwarf plants. So I'll mention it here. First of all, you need to keep in mind that first thing here is that Mendel cross-pollinated homozygous tall plants with homozygous dwarf plants. Okay, so here what are the gametes? Which gametes will you observe here? Capital T, small t, and small t. Okay, so now let us perform a cross between them. Okay, so what are we going to observe in this? Let us check. I'll change the color. And so what are we going to observe here is this. Again, this. We are crossing this, okay? Then capital T, small t. Then again, capital T, small t. Now, what do you observe in this? All the gametes, that is, all the FN generation are tall. Okay, I repeat, Mendel cross-pollinated homozygous dwarf plants with homozygous tall plants. Now, what did he observe? These were the parental generations, the ones that you see on the top. Now, then the seeds produced by cross-pollinated, that is P generation plants, they were collected. Now, these P generation plants were collected and they were grown. Now, these plants, they all were hybrid. Why are they hybrid? Because they all are heterozygous. I hope you remember the terms now, homozygous, heterozygous. They all are hybrid because now they all have these two genes, but the dominant one is the tall one, right? That is capital T. So that is why all the plants were tall. So basically, can we say that F1 generation, all the plants were tall? Yes? Okay. Then when we move ahead in this, now what he did is all of these F1 plants, all of these F1 generations were allowed to self-pollinate now. They were all allowed to self-pollinate. Now what is going to happen now? I think you all must be predicting it all already. Now in this case, because we are self-pollinating the plants which came from the F1 generation only. So what are we going to take here? We will mention the same plants that had come from the F1 generation. So we will mention here these particular gametes. Yes, you got it right. Okay, now we'll put them here. I hope it's very very conceptual and very easy, and at the same time these questions are very, very scoring because there is no scope for you to get it wrong if you've understood it. There is nothing that you'll forget in this, I'm sure. So what are you going to get here? Capital T, then you'll get here capital T, small t. Then again, you will get here capital T and small t. And the last one are both small t. Now you tell me, what is the difference that you see here? Now he noted the 75%, okay, plants of the F2 generation were tall and 25% were dwarf. So this shows that now in F2 generation, you don't have all the plants tall. What is the ratio you see here? There are 3 tall plants and 1 dwarf plant. So as per the physical appearance, the phenotypic, I hope now you remember what is phenotypic and genotypic. The phenotypic ratio here you are going to get as 3:1. Why? Because you can see here that these 3 are tall. And what are we going to write here? What do you see here? Yes, here we can check. I'll just write it here once. One I will write and then I'm sure you'll be able to get it. This is homozygous. Homozygous. And this is tall. This is also, but these two are heterozygous. Both of them. Okay. And same is the case with this also. This is again homozygous. Okay. But now we're going to check the genotypic ratio. Genotypic ratio. Now what are we going to get now? See, this one is completely, all are dominant in this. T and T both. The both are capital T, right? So this is one. Second one, here there are two which are heterozygous. So here there are two which have these particular genes. So you'll take it two. And only one is there in which both are recessive. So here you will get as one again. So what did he observe here? That F2 generation, the tall and dwarf plants were produced in the ratio of 3:1. And then he raised again F3 generation from F2 plants, again by self-pollination. So what basically will you observe here? The phenotypic ratio will be 3:1 and genotypic ratio will be 1:2:1. Okay, so what is the conclusion? That all of these experiments showed a similar pattern of inheritance, isn't it? One of the contrasting features, that is dwarf case. First one, let us check this one again. Once again, come here. The first one, what do you observe? That dwarf in this case did not appear at all, isn't it? All the plants were tall. And this contrasting feature reappeared in the F2 generation. Here, dwarf was not there, and again they reappeared in the F2 generation. So both the features were found approximately in what ratio? 3:1. But here all were tall and there was no dwarf plant observed. Okay, so I hope this particular cross is clear to you. And apart from this, then we'll be talking about some laws in the next session. Before that, just go through this once and I'll be posting some questions on the community post. Try to solve it and we'll see you in the next class. So take care and have a nice day. I hope you have understood. Please don't forget to subscribe and don't forget to like if you've understood. And in case you have certain queries, you can mail us queries or suggestions. You can mail us on chemistrybiologyfacts2511@gmail.com. Thank you and have a nice day.